Subscribe to our channel and press the bell icon so that you never miss any video lesson from RAU'S IAS Study Circle.
All of you who are preparing for the civil service examination should understand the importance of essay paper and pay attention to this. First, this paper is of 250 marks and its weight is around 12.5% in your written plus interview score. This is a significant portion and hence you should prepare this very seriously. Second important aspect is average score of GS paper of the candidates who actually clear the examination hovers from around 95 to 105 marks. At the same time, average marks of essay for these people is around 130 plus. Toppers usually score 140 plus marks in this paper and take a lead over the competition. Third, essay paper develops on your foundation of general studies, your knowledge and understanding of various subjects, ability to present your ideas lucidly, covering a wide spectrum of interrelated ideas, and inherent skill for presenting your ideas in a systematic and powerful manner helps you in reaping more benefits from your general studies. In order to help you prepare for the essay paper in a systematic and gradual manner, RAU'S IAS has started Mains Essay Test Series and QIP program. The first class of the session will be an open session that all of you can join. The session will take place on 12th of October at 10:30 am. There is a link in the description through which you can register yourself for the session. Attend the session and ensure a better chance for clearing the mains examination.
Hello and welcome to Daily News Simplified, an answer to what, why and how of newspaper reading from our UPSC perspective. The news to be discussed has been displayed on your screen and the timestamping for the same has been provided in the description box below. So on this note, let us understand the important news which has appeared in the New Delhi edition of the Hindu newspaper. The first news to be discussed appears on page number 14 with respect to this year's Nobel Peace Prize. The news says, leading the global fight against hunger, World Food Programme. So this year, the Nobel Peace Prize has been given to World Food Programme. And it is in this regard, this news says that UN humanitarian organization which won this year's Nobel Peace Prize says there won't be a peaceful world as long there is hunger. So one of the most important part with respect to giving World Food Programme the Nobel Peace Prize is the linkage or the relationship between hunger and armed conflict, which basically refers to the fact that armed conflict as well as war, including violence, create a situation of hunger as well as food insecurity. And if armed conflict continues in a particular area for a sustained period of time, then such armed conflict definitely leads up to hunger or lack of food, or we can say that there is food insecurity in such regions. Now, food insecurity in such regions can also flare up the conflict in any region which is deprived of food. So basically, food insecurity can also lead to violence. So it is in this regard the Nobel Committee believes that food security in any region promotes stability as well as peace. And it is because of this World Food Programme has been bestowed the Nobel Peace Prize for 2020. Now the World Food Programme was established in the 1960s during the Kennedy administration of United States. Now the backdrop to establish World Food Programme was because of the devastating impact of World War II, especially on children because of hunger. Now, it was seen that because of the World War and the devastating impact the Second World War created, lots of children as well as people went hungry. So it was realized that a multilateral forum or an organization is needed which can supply food to such areas of armed conflict. And it is here where the role of World Food Programme becomes very important, as it has played certain very important roles in our history in order to provide food security, especially in areas hit by crisis. So one of the major efforts of World Food Programme is towards achieving zero hunger, as it is also one of the Sustainable Development Goals, or rather we can say it is the SDG 2. So we can say that the effort of World Food Programme in reducing hunger also in a way helps to achieve the Sustainable Development Goal 2, that is the target to achieve zero hunger. Now there is another important aspect here, that hunger also leads to malnutrition. And malnutrition, especially among children, further reduces their productivity. So we can say that the World Food Program not only reduces hunger but also improves nutrition among such hungry people, especially the children. And another important aspect which we have just seen, the relation between hunger as well as armed conflict. Now, just after the creation of World Food Program in 1962, northern Iran was hit by a very severe earthquake, and it was here where the mantle of World Food Program was established, as it provided food to thousands of people who were rendered homeless because of the devastating earthquake. Now, further, the World Food Program played key role in providing food security to specially areas hit by different types of crisis. For example, because of the famines in the Sahel region in the 1970s, World Food Program played a major role in providing food to such region. It also played a major role with respect to famine in Ethiopia, with respect to conflict in southern Sudan, tsunami which took place in 2004 in the Indian Ocean, and also with respect to earthquakes in Haiti and Nepal very recently. So the news highlights here with respect to World Food Program that by the year 1965, the experimental agency with an initial mandate of 3 years had proved its worth to the world after responding to multiple crises and was enshrined as a fully fledged UN program. And it is in this regard it stated that it is to last for as long as multilateral food aid is found feasible and desirable. Now, the World Food Program also played a major role with respect to various disease outbreaks, including Ebola in Africa and also COVID 19 in recent times. Now, since we have mentioned about the Sahel region, you must know with respect to the world map as far as prelims as well as mains is concerned. So, as you can see in this map, this is the region of Sahel in Africa. And as you can see, it forms a connection or the linkage with respect to Sahara Desert in the north, as well as more fertile and tropical regions in the south of Africa. So in this regard, you must know about the Sahel region which is in Africa. So here it highlights that it is a largely semi-arid belt of barren, sandy and rock-strewn land. The Sahel marks the physical and cultural transition between the continent's more fertile tropical regions to the south, that is these regions, and its desert in the north. So here there are two important aspects with respect to the Sahel region, that it marks as the physical and cultural transition between two parts of the continent, that is with the more fertile tropical region in the south and with the Sahara Desert in the north. So here it says that commonly the Sahel stretches from Senegal on the Atlantic coast through parts of Mauritania, Mali, Burkina Faso, Niger, Nigeria, Chad and Sudan to Eritrea on the Red Sea coast. Further, it highlights that culturally and historically, the Sahel is the shoreline between the Middle East and the Sub-Saharan Africa. Now, this means that it is the site of interaction between Arabic, Islamic, and nomadic cultures from the north and indigenous and traditional cultures from the south. So, this region of Africa, that is Sahel, becomes important, especially from the perspective of world geography. Now, the World Food Program has devised certain special initiatives, especially for the Sahel region. It says that the overarching goal of the UN support plan for the Sahel is to scale up efforts to accelerate shared prosperity and lasting peace in the Sahel countries. So again, we see a relation with respect to providing food as well as peace and stability in the region, and thereby at large implementing priorities to achieve the 2030 Agenda for Sustainable Development and the African Agenda 2063. So again, all these aspects becomes important from our examination perspective. Now the news also highlights about the widening role of World Food Program, especially with respect to not only providing food in times of emergency or crisis, but also addressing the nutritional requirements. So here the news highlights that the organization has widened its operational remit and is now a leading provider of not just emergency food aid, but also an agency engaged in supporting the nutritional requirements of communities through food assistance programs. Further, it says that the organization has also owned its response capabilities to the point where it is able to serve as the frontline telecommunication and logistical support provider to all UN agencies and NGOs in any crisis situation. So the World Food Program also provides logistics support to all United Nations agencies as well as NGOs in times of crisis. It says that from Ebola outbreak in West Africa to the Nepali earthquake of 2015. So we see that the role of World Food Programme has widened over a period of years, and it is because of this the Nobel Peace Prize for the year 2020 has been awarded to the World Food Programme. So the Nobel Committee highlights that the world is in danger of experiencing hunger crisis of inconceivable proportions if the World Food Programme and other food assistance organizations do not receive financial support they have requested. So here there is also need to provide financial support to World Food Programme. And it is here also the Nobel Peace Prize Committee has highlighted or rather stressed or emphasized the linkage between hunger and armed conflict. It says that the link between hunger and armed conflict is a vicious circle. War and conflict can cause food insecurity and hunger, just as hunger and food insecurity can cause latent conflicts to flare up and trigger the use of violence. Now we have already seen this particular aspect. It says that we will never achieve goal of zero hunger unless we also put an end to war and armed conflict. So it is here where the role of World Food Programme becomes important in addressing the issue of food insecurity, especially in areas affected by armed conflict. So with respect to this relation between armed conflict and zero hunger, the Norwegian Nobel Committee has emphasized that providing assistance to increase food security not only prevents hunger but can also help to improve prospects for stability and peace. And it is here where the Nobel Committee believes that the World Food Programme as an organization has taken the lead in combining humanitarian work along with peace efforts through pioneering projects in South America, Africa, as well as Asia. Now, the World Food Programme was also an active participant in the diplomatic process which culminated in adoption of United Nations Security Council's Resolution 2417. Now, Resolution 2417 for the first time explicitly addressed the linkage between conflict and hunger. So the Resolution 2417 of United Nations Security Council addresses the issue of conflict and hunger, especially with respect to humanitarian impact. It says that the Council drew attention to the link between armed conflict and conflict-induced food insecurity and also the threat of famine. So there are three important aspects highlighted here. So these 3 points are with respect to the relation between armed conflict and food insecurity and also the threat of famine in the region. Further, the Security Council resolution also stressed on the fact that armed conflicts as well as violation of international law and related food insecurity could be drivers of forced displacement. So, because of armed conflict induced food insecurity, it can lead to forced displacement of people from their home, and this is also one of the points highlighted or stressed by this particular resolution of United Nations Security Council. So the Council says that it strongly condemned the unlawful denial of such access, that is access to food, and depriving civilians of objects indispensable to their survival, that is food, including willfully impeding relief supply and access for responses to conflict-induced food insecurity. So again, from our examination perspective, we need to know about the United Nations Security Council Resolution 2417, especially with respect to conflict and hunger. And also because of the fact that World Food Programme has now received Nobel Peace Prize. So for this year's award, the Nobel Committee wishes to turn the eyes of the world towards the millions of people who suffer from or face the threat of hunger. Now, here one important thing to understand is that food insecurity as well as world hunger becomes one of the important topic with respect to our UPSC mains examination and also with respect to our essay. As questions may be asked with respect to food insecurity not only in the world but also in India or in certain parts of the country, and also this aspect can be asked as a part of your essay. So overall, the Nobel Committee highlights that the World Food Programme plays a key role in multilateral cooperation on making food security an instrument of peace and has made a strong contribution towards mobilizing UN member states to combat the use of hunger as a weapon of war and conflict. So again, we need to understand the correlation between providing food security as an instrument of peace. So this is a very important message for the world, especially to achieve Sustainable Development Goal 2 of zero hunger. Now from our prelims point of view, this topic gets covered under current events of international importance as well as sustainable development and poverty. And from our mains point of view, it gets covered under GS paper 2 as well as GS paper 3. Now in GS paper 2, it specifically gets covered under issues relating to poverty and hunger, and also important international institutions, agencies and forum including their structure and mandate. And in GS paper 3, it gets covered with respect to food security as well as public distribution systems, especially with respect to India. Thus, this news becomes very important both from our prelims as well as mains perspective. And with respect to World Food Organization, please remember SDG 2 goals of achieving zero hunger. With this, let's move on to our next news of discussion. The next news to be discussed appears as a FAQ on page number 13. It says, Genetic Scissors and Rewriting the Code of Life. How does a method for genome editing developed by two women scientists help in tackling the diseases? Now, the technology with respect to gene editing in news is CRISPR-Cas9 technology, which is a genetic scissor. Now, the CRISPR-Cas9 technology of gene editing is being used to find cure for diseases such as ALS, which is a motor neuron disease, blood disorders, as well as COVID. And because of the success of CRISPR-Cas9 technology as a gene editing technology, the Royal Swedish Academy of Sciences has decided to award the Nobel Prize in Chemistry 2020 to Emmanuelle Charpentier as well as Jennifer A. Doudna. And both these women have been awarded the Nobel Prize in Chemistry for 2020 for the development of a method for genome editing. So in this FAQ, let us understand about the CRISPR-Cas9 technology as well as various applications of CRISPR-Cas9 technology, as this topic becomes important not only with respect to UPSC prelims examination but also with respect to upcoming mains examinations, especially with the use and applications of CRISPR-Cas9 technology with respect to gene editing under GS paper 3. So in this regard, first of all, let us understand certain basics with respect to gene, DNA as well as chromosome. Now, chromosomes are found in the nucleus of a cell. Now, with respect to chromosome, it comprises of DNA. And as you can see that these genes are part of DNA. So, it highlights that nucleus contains thread-like structure called chromosomes. So, chromosome is found within the nucleus of a cell. Further, it says that each chromosome is made up of tightly coiled strands of deoxyribonucleic acid. That is, each chromosome is made up of DNA. And genes are segments of DNA that determine specific traits of an individual or any species, or color of an eye, or even color of a hair. Further, it highlights that chromosomes carry genes and help in inheritance or transfer of characters from the parents to the offspring. And in this regard, genes can be considered as unit of inheritance in living organism. So overall, genes determine the transfer of genetic characters or hereditary characters from parents to offspring. So overall, we can say that DNA contains or comprises the hereditary materials or the genetic materials in all living beings. Further, with respect to DNA, it highlights that the information in DNA is stored as a code made up of 4 chemical bases. Now, these 4 chemical bases are adenine base, guanine base, cytosine base, as well as thymine base. Now, for more clarity, these are the different bases in a DNA. And these different bases are also referred as nucleotide base pairs, that is thymine, adenine, cytosine and guanine. Now, as you can see, these different nucleotide base pairs appears as steps of a ladder. Now, the pairing of these nucleotide base pairs takes place in the form of A with T and C with G, that is adenine with thymine and cytosine with guanine. So it says that DNA bases pair up with each other, that is A with T and C with G, to form units called base pairs. And each base is also attached to a sugar molecule and a phosphate molecule. And together, that is a base pair, that is a nucleotide, a sugar molecule, as well as a phosphate molecule are called nucleotides. So as you can see here in this particular picture, nucleotide consists of a base pair, a sugar molecule and a phosphate molecule. Another important aspect is that nucleotides are arranged in 2 long strands that form a spiral called a double helix. And the structure of the double helix is somewhat like a ladder with the base pairs forming the ladder's rung or the ladder's step. Something like this particular picture. So it says that the structure of double helix is somewhat like a ladder with base pairs forming the ladder steps and the sugar and phosphate molecules forming the vertical side pieces of the ladder. To have more clarity, these sugar and phosphate molecules are on the side part of the DNA. So these are some of the important information with respect to gene, DNA as well as chromosome. Now these basic knowledge is important to understand how the CRISPR-Cas9 technology functions. So the CRISPR-Cas9 technology provides for or helps in adding, altering or even deleting the genomic code in living beings. Now what this technology does is to find a specific bit of DNA inside a cell. Now that specific bit of DNA may be an injured DNA or maybe a foreign DNA. After that, the next step in CRISPR-Cas9 technology of gene editing is usually to alter that piece of DNA or to remove that piece of DNA. However, there are other aspects as well for which the CRISPR-Cas9 technology has been used for or adapted to, such as turning genes on or off, which means to say is that every gene has certain functions. So turning genes off means not to use that particular gene in a living being. And the turning off of gene is done without altering their sequences. So gene editing also allows for gene manipulation. So all these aspects of gene editing, gene manipulation or even gene deletion has various potential applications, including correcting genetic defects or genetic disorders, treating as well as preventing the spread of diseases and also improving crops. So these can be said to be some of the applications with respect to use of CRISPR-Cas9 technology. Now coming to another important question is that how does this technology work or functions? Now, as we have already seen that the CRISPR-Cas9 technology edits a part of gene. So how it does that? It says that CRISPRs are specialized stretches of DNA and the protein Cas9 is an enzyme found in bacteria that acts like a pair of molecular scissors capable of cutting strands of DNA or cutting parts of DNA. Now the technology on which CRISPR-Cas9 functions is almost similar to the functionality of how a bacteria protects itself from attack from a virus. Now we have already seen that Cas9 is a protein found in the bacteria and this is how the CRISPR-Cas9 technology functions. So the technology of CRISPR-Cas9 functions in the same way bacteria responds to virus. Now here let us understand how a bacteria responds to virus. So when a bacteria is attacked by a virus, such bacteria captures snippets or parts of DNA from the invading virus. So suppose if this is the DNA of the virus, then the bacteria captures certain part of the DNA of the virus. Suppose this part of the DNA of the virus is being captured by the bacteria. Now the bacteria uses the part of the DNA which it captures of the virus to form DNA segments, and this DNA segments comprises of CRISPR arrays. Now this CRISPR arrays basically helps the bacteria to remember the traits of the virus, and this CRISPR array is stored in the bacteria. Once the bacteria is attacked by a virus, then it uses a part of the DNA of the virus to form DNA segments, and this leads to creation of CRISPR arrays within the bacteria, and this CRISPR array helps the bacteria to remember the traits of the virus. So basically what happens here is that suppose this virus attacks the bacteria again. So what the bacteria does is that bacteria again produces these RNA segments from the CRISPR arrays which it had stored earlier to target the DNA of the virus and thereby destroys the virus. And it is this methodology which is imbibed in the CRISPR-Cas9 technology of genome or gene editing. So in this regard, this news highlights that CRISPR-Cas9 was adapted from a naturally occurring genome editing system in bacteria. The bacteria captures snippets of DNA from invading viruses, and then the bacteria use these snippets of DNA to create DNA segments known as CRISPR arrays. So the DNA segments which the bacteria creates by capturing snippets of DNA from the virus is also referred as CRISPR arrays. It says that the CRISPR arrays then allows the bacteria to remember the virus or closely related ones or associated virus. And if the virus attacks again, the bacteria produces these RNA segments from the CRISPR arrays to target the virus DNA. So it says that the bacteria then use Cas9 or a similar enzyme or protein to cut the DNA apart which disables the virus. And the same technology or methodology is adopted by CRISPR-Cas9 technology. So both Nobel laureates used this understanding of protection by bacteria to develop the Cas9 technology for gene editing. Now here the news highlights about a tracer RNA or trcrRNA. Now let us understand few basic aspects here. So this news highlights that Emmanuel, while studying about a species of bacteria known to be associated with a range of illness such as pharyngitis, tonsils, scarlet fever, etc., she discovered a previously unknown molecule, tracer RNA. And her work showed that this tracer RNA is a part of bacteria's ancient immune system, that is CRISPR-Cas, that disarms the virus by splitting their DNA. And based on this understanding, Emmanuel initiated a collaboration with biochemist Jennifer Doudna. Now, together, these two ladies succeeded in recreating the bacteria's genetic scissors in a test tube and simplifying the scissors' molecular components so that they were easier to use. Now, to understand this entire thing in a very layman or very simple way, there are two aspects. First is the identification of the area where gene editing is required. So the question is, how will we identify such area? The second is the editing of the DNA or the gene itself. So these are the two aspects which became very important with respect to the CRISPR-Cas9 technology. So this news highlights that the CRISPR-Cas9 system relies on two main components, a guide RNA and a CRISPR-associated Cas nuclease. Now here very simply let us understand about the role of guide RNA. Now guide RNA recognizes the target region where DNA is to be edited. And once the target area is identified, then the guide RNA sends the Cas9 nuclease for gene editing. So this is how basically CRISPR-Cas9 technology functions as. Now with respect to guide RNA as well, there are two aspects. It comprises of a CRISPR RNA and a tracer RNA. Now here it's important to understand that the tracer RNA serves as a binding scaffold to the Cas nuclease, which is sent by the guide RNA for gene editing. Thus, from an examination perspective, you need to know about the role played by guide RNA, CRISPR-associated Cas nuclease, as well as the two components of guide RNA, that is CRISPR RNA as well as tracer RNA. So these aspects becomes important from the perspective of general science in the prelims examination as well as science and technology with respect to your mains examination. Now there was another significant step which both the Nobel laureates achieved. It says that in a significant experiment, both of them reprogrammed the genetic scissors. In their natural form, the scissors recognize DNA from the viruses. However, both the Nobel laureates proved that such DNA from virus could also be controlled so that they can cut any DNA molecule at a predetermined site. So through their experiment, both the Nobel laureates proved that the virus can be controlled by cutting or editing the DNA at a selected location or at a recognized location. So the CRISPR-Cas9 technology as compared to other gene editing systems is much more easier as well as much more targeted to use. Further, apart from gene editing, this technology also allows researchers to find out what genes do, study about their mutations associated with disease, and also test for new treatments including various combinations or mutations with respect to editing of the gene or the DNA. Now with respect to the use of CRISPR-Cas9 technology, it is being used to treat ALS disease or the motor neuron disease, blood disorders, even COVID-19. Now the blood disorders include beta thalassemia as well as sickle cell disease. So this news highlights that earlier this year a person with hereditary blindness became the first person to have a CRISPR-Cas9 based therapy directly injected into her body. Another benefit of using CRISPR-Cas9 technology is that with respect to beta thalassemia as well as sickle cell disease, that is both blood related disease, blood transfusions would no longer be required because such persons' bone marrow stem cells were edited using CRISPR-Cas9 technology. So again, the benefit we see here is that with respect to blood related disease, blood transfusion will not be required. Now as already stated that beta thalassemia is a blood disorder that reduces the production of hemoglobin. Now, hemoglobin is important for the body as hemoglobin carries oxygen to the cells throughout the body. And in people with beta thalassemia, low levels of hemoglobin lead to a lack of oxygen in many parts of the body. And in the present time of COVID-19, lack of hemoglobin also is very dangerous. Further, the CRISPR-Cas9 technology also helps to treat people with sickle cell disease. Now, sickle cell disease is again a blood-related disorder or a group of inherited red blood cell disorders which affects the hemoglobin. Now, here it highlights that normally the red blood cells are disc-shaped and flexible to move easily through the blood vessel. Now, in sickle cell diseases, these cells do not bend or move easily and therefore can block the blood flow to the rest of the body, and that's why it is very dangerous. So, again, without any blood transfusion, both beta thalassemia as well as sickle cell disease can be cured using CRISPR Cas9 technology. Now, work is also being carried on to treat the motor neuron disease, which is also referred as ALS, that is amyotrophic lateral sclerosis. Now, ALS is a group of rare neurological disease that mainly involve the nerve cells or the neurons, which is responsible for controlling voluntary muscle movement. So, the voluntary muscle movement, such as chewing, walking, and talking, does not take place. And if you remember very correctly, the famous scientist Stephen Hawking had ALS disease. And this was very well depicted in the movie Theory of Everything. Now, motor neurons are nerve cells that extend from the brain to the spinal cord and to the muscles throughout the body. Now, these motor neurons initiate and provide vital communication links between the brain and the voluntary muscles. Hence, when a person is diagnosed with ALS, then it results in gradual deterioration or death of motor neurons. Further, Dr. Doudna is also employing the CRISPR-Cas9 technology in battling COVID-19 to develop a test to detect infections of COVID-19. Now, two important aspects needs to be understood here. One is with respect to Feluda and the next is with respect to biohacking. Now, the CSIR Institute of Genomics and Integrative Biology in Delhi have developed a COVID-19 testing kit which they have nicknamed Feluda or FNCAS9 Editor-Linked Uniform Detection Assay. And this Feluda mechanism is based on CRISPR-Cas9 technology. Another important aspect of CRISPR-Cas9 technology is its use in biohacking. Now, biohacking involves a range of activities where a person either develop themselves or improve their habits or uses one gene or hacks one gene for further evolution, or also biohacking can be used to develop transhuman. Now, it is here where the ethical concerns about use of CRISPR-Cas9 develops. Thus, this news becomes important to understand the role of CRISPR-Cas9 technology, which is used for the development of genome editing, and because of which Emmanuel Charpentier and Jennifer A. Doudna has been awarded the Nobel Prize in Chemistry for the year 2020. And this topic becomes important with respect to general science in the prelims examination and in mains gets covered under GS paper 3. With this, let's move on to our next news discussion. The next news to be discussed appears on page number 8. It says Navy to hold PASSEX with United States carrier. Now, PASSEX here refers to passage exercise with United States Navy aircraft carrier group, which will take place on 12th of October. So the news highlights that the Navy ships of India will hold a PASSEX, that is passage exercise, with United States Ronald Reagan on 12th of October, that is tomorrow, as the United States ships transit through the Indian Ocean. So it highlights that the PASSEX is likely to be held in the waters of Kochi, near the Indian Ocean. Now, PASSEX, or a passing exercise, is an exercise done between two navies to ensure that navies are able to communicate and cooperate in times of war or humanitarian relief. Now, the whole idea of conducting PASSEX is to practice cooperation between two navies. However, there can be other unofficial reasons as well, including the show of power of a nation through public display of naval power by showing the flag of the particular navy, or even for other political or diplomatic reasons. So, considering this backdrop, PASSEX between India and United States becomes important. Further, the news mentions that on 25th September, for the first time, a United States Navy P-8A long-range maritime patrol aircraft landed at Port Blair, Andaman and Nicobar Islands, and it landed for refueling under the Bilateral Logistics Support Agreement. So again, the Bilateral Logistics Support Agreement between India and United States becomes important here. Further, between India and United States, in July 2020, frontline warships of the Navy conducted PASSEX, that is passage exercise, with US Navy Nimitz carrier strike group near the Andaman and Nicobar Islands as it was transiting the Indian Ocean. Now, the US Nimitz was returning from South China Sea through the Strait of Malacca, where it undertook the freedom of navigation operations. Now, freedom of navigation operation are one of the principal tools by which United States challenges maritime claims deemed excessive under international law. Now, here the reference is obviously with respect to China, as Nimitz was carrying out the freedom of navigation operation in the South China Sea. Now, further in August 2020, the United States Ronald Reagan was in the South China Sea to conduct freedom of navigation operations amid tensions between China and the United States. So we understand that US is carrying on freedom of navigation operations in the South China Sea as a part of its exercise to challenge excessive Chinese aggression. Further, the news says that a trilateral Malabar naval exercise with Japan and United States is also scheduled to be held in November, though there is no clarity on inviting Australia for the exercise. So, here the point to be understood is that if Australia is invited to the Malabar trilateral exercise, then this invitation to Australia completes Quad, and this is what United States is looking forward to for Quad as a military alliance to conduct joint navy operations. So again, let's see whether India invites Australia or not for the upcoming Malabar naval exercise. Now in this news, it highlights about Strait of Malacca. So from our examination perspective, we need to know about the location of Strait of Malacca. Now as you can see in the world map, this is where the Strait of Malacca is. And with respect to this map, we see that it is close to Singapore and it separates Indonesia as well as Malaysia. Thus, this topic becomes important with respect to current events of national and international importance, security, as well as world geography. So, with this discussion, let's move on to our next news analysis. The next news to be discussed appears on page number 5. The news says, shrinking spaces for winged visitors a cause for concern. On World Migratory Bird Day, environmentalists and experts say habitat for flamingos and wetland birds has shrunk 20% of what it was. Now, this news basically stresses about the importance of protection for lesser flamingos because of destruction of wetlands along with developmental activities such as development of Navi Mumbai Airport, Mumbai Trans Harbour Link, as well as real estate projects have left very little space for the flamingos. So it is in this regard the environmentalists in Mumbai have expressed their disappointment, especially with respect to shrinking of area available for flamingos. Now, this news highlights that on World Migratory Day, environmentalists and experts say habitat for flamingos and wetland has shrunk to 20% of what it was. So, in this regard, we also need to know about the World Migratory Bird Day. So, the World Migratory Bird Day is an annual awareness raising campaign which highlights the need for the conservation of migratory birds as well as their habitats. Further, the World Migratory Bird Day has a global outreach and is an effective tool to help raise global awareness with respect to the threats faced by migratory birds, their ecological importance, as well as the need for international cooperation for their conservation. And it is in this regard this year's the theme for World Migratory Bird Day is Birds Connect Our World. And this was chosen to highlight the importance of conserving and restoring the ecological connectivity and integrity of ecosystems that support the natural movements of migratory birds and also which are essential for their survival and well-being. So basically this news highlights about the fact that space for flamingos is retreating or shrinking in Mumbai. And in this regard, government should not destroy or destruct more wetlands in the Navi Mumbai region. Further, this news also mentions about World Migratory Bird Day. So, the World Migratory Bird Day is an annual awareness raising exercise or campaign which highlights the need for conservation of migratory birds and their habitats. And for this year, the theme of World Migratory Bird Day is Birds Connect Our World. Now, another important fact which you must know is that flamingos are protected under Schedule 4 of the Wildlife Protection Act of 1972. And you must know about Thane Creek Flamingo Sanctuary, which has been created near Mumbai for the protection of flamingos. Thus, this news becomes important with respect to general issues on environmental ecology, biodiversity, as well as climate change. Now, after our discussion, let us take up today's question for the day. The question is, consider the following statements. First, CRISPR arrays helps the bacteria to target DNA of virus. Second, gRNA plays a major role in editing gene. And third, bone marrow stem cells can be edited by CRISPR-Cas9 techniques. So the question is, which of the statements given above are correct? Options are A, 2 and 3 only, B, 1 and 2 only, C, 1 and 3 only, and D, 1, 2 and 3. Now coming to the answer of yesterday, the question was, which of the following statements are correct? Options were, India is the largest producer of cotton in the world, and second, India has the largest area under cotton cultivation in the world. So the question was, select the correct answer using the code given below. Here the correct answer is C, that is both 1 and 2. So with this, we come to an end to today's discussion. Thank you.